Hello, everyone. So my name is Allison. I am a PhD candidate at Western University, and I am from the Genomics platform. And so what I'm going to be talking to you about today is investigating genetics' role in all of the neurodegenerative diseases we're looking at. So what is genetics? Well, it's the study of individual genes and how they're passed down from parents to children. And so you get all of your DNA from your biological mother and your biological father. And we study how that kind of happens and what that DNA looks like. So when you think of the human body, it's made up of billions and billions of cells. And inside each one of your cells is a copy of your DNA. So this spiral represents your DNA. And it's the code that makes up you. You can think of it as a very complicated instruction manual. And so within your DNA, there are approximately 20,000 different genes. And each of those genes encodes a protein. And proteins are essentially the building block of life. The proteins come together, they make up the cells, which make up the tissues, the organs, and you. And so, when you, our participants, were enrolled into ONDRI, you went into a Life Labs location, and you gave a blood sample. And that's probably as much as you really knew about the genomics platform. Well, that blood sample was sent to my lab. And at my lab, we carefully extracted the DNA. And with that DNA, we sequenced it. And from that sequence, we looked for something called variants. So your DNA sequence is in the form of, and I don't have a laser pointer, but it's in the form of A's, C's, G's, and T's. And so we looked at that sequence, and we looked for changes in the sequence. We also carefully stored all of this DNA sequencing and stored the DNA itself. So what is a DNA variant? Well, we have a basic idea of what a normal sequence of DNA would look like for a human. And so we take a participant's DNA sequence, and we compare it to that normal. And any sort of change within the DNA is called a variant. They can be very common in the general population. They can be very rare in the general population. But any sort of change we keep track of. And so it could be a substitution of one letter to another letter. For example, here we have an A to a G. It could be an insertion of letters that aren't usually there. So here we have two T's that aren't really in the normal sequence. Or it could be a deletion. So here we have a deletion of two A's and a G. And these changes can be of various sizes. They can be one base or one letter, or they could be many, many letters, thousands of letters. So specifically for ONDRI, what we did is we custom designed a special tool called ONDRI-seq, and it actually only looks at 80 of those 20,000 genes that I mentioned previously. And so the reason we only looked at 80 genes was because there's already a subset of genes that previous researchers have looked at and said, we think these are associated with neurodegenerative diseases. And so to be very efficient, we wanted to look at those 80 genes specifically. And so, as I said, each of these genes has already been somewhat associated with one of the diseases that ONDRI covers. So for the purposes of this talk, I'm going to introduce you to one gene, and in specific, one variant. So that gene is called ApoE, and the protein it encodes is apolipoprotein E. And what apolipoprotein E does is it carries fats throughout the brain. It has many other roles too, but when there's a variant within ApoE, it's thought that it becomes less efficient, and at a protein level, it contributes to these neurodegenerative diseases, specifically Alzheimer's disease. So as you can see here, we have an E3, an ApoE E3, and an ApoE E4. So ApoE E3 is what we would call the normal, as I kind of showed you a few slides back. But if there's a variant within ApoE at position 130, we get the ApoE E4 variant. And ApoE E4 has been associated with an increased risk of Alzheimer's disease. Now, it's important to note that this is just a risk factor. If someone has the ApoE4 variant, it doesn't mean they're going to get Alzheimer's disease. It just means they're at an increased risk compared to someone who has the E3. It's also important to note there's actually another version of ApoE called ApoE2, which I won't get too much into for this talk, but it's actually thought that it's protective against Alzheimer's disease. And so it kind of does the opposite of ApoE4. So there's variants that can increase your risk for neurodegeneration or many diseases, and there's also variants that can protect you. So going back to ApoE4, as I noted earlier, in genetics, when we study genetics, it's important to know that you get DNA from both your mother and your father. So you have 2 copies of every gene. So you could have 2 copies of the normal ApoE, ApoE3, or you can have copies of the ApoE4. So here we have an ApoE4 heterozygote, and what that means is you have 1 copy of ApoE3 and 1 copy of ApoE4. So you have a moderately increased risk of developing Alzheimer's disease. If you have 2 copies of ApoE4, then we call you a homozygote, and you have a slightly more increased risk of developing Alzheimer's disease. And so because of this risk association, researchers are really interested in whether ApoE is also associated with other neurodegenerative diseases. And with ONDRI, we have a really unique opportunity that we can look at all of those diseases at the exact same time. So that's what we did. And we saw that, as we expected, the Alzheimer's and mild cognitive impairment cohort had an increased percentage of participants that have at least 1 copy of the ApoE4 variant. And you can see we did run some controls on this test, and so the Alzheimer's participants, there's approximately 50% of participants with at least 1 copy, whereas only 25% of our controls had only 1 copy. None of the other cohorts had a significantly increased frequency of participants with the E4 allele. And you can kind of see that here again, or the E4 variant. You can kind of see that here again. So here I looked at people with 2 copies of the ApoE4 variant. And as you can see, we have many more participants in the Alzheimer's cohort with 2 copies. And so these results do 2 different things. They validate the diagnoses of our Alzheimer's mild cognitive impairment participants in a way, because we can look at the frequencies that have been seen in different groups of Alzheimer's participants in the past for different studies, and we're seeing very similar frequencies of the E4 variant. This is also one of the first studies to look at the ApoE E4 variant across all 5 of these diseases at one time using all the same enrollment criteria, and ONDRI really gives a unique perspective on these genetic studies. So we can continue with ApoE. What we'll be doing is we'll be looking at if this ApoE4 variant is influencing actual symptoms of neurodegeneration. So just because it's not doesn't seem to be contributing to the Parkinson's, for example, presentation, it might be contributing to a presentation within Parkinson's disease. So for example, I'm currently working with the neuropsychology platform looking at measures of cognitive impairment to see if the ApoE4 variant is causing a increased cognitive impairment burden across the neurodegenerative diseases. And then we also have 79 other genes to look at the variation within those genes and do very similar studies. And so the importance of the research, well, it'll get us a better understanding of what could be contributing to the diseases on a gene-protein level, so a really small scale within the brain. We can also identify new targets for treatments, and very importantly, this work might allow us to develop a new DNA sequencing-based diagnostic and risk assessment tool, which is something we're currently working on. And importantly, as everyone else has done before, I want to thank all of you. Without you, this research obviously wouldn't be possible. And thank you to all the other ONDRI scientists and clinicians as well.